truly my great pleasure to introduce to you a distinguished scholar and researcher, Dr. Alessandro Gozzi. Dr. Gozzi is a senior scientist and director of Functional Neuroimaging Laboratory at the Italian Institute of Technology in Rovereto, Italy. Dr. Gozzi's lab focuses on studying functional organization of mammalian brain at macro scale. A major goal of Dr. Gozzi's research is to unravel the neural basis and dynamic organization of large-scale functional connectivity as a measure with fMRI. And the underpinning of these disruption is disruption in a developmental disorder like autism. Dr. Gozzi's lab addresses this question via a combination of cutting-edge functional MRI mapping with genetic and neural manipulation in the living mouse brain. This approach has laid the foundation for a novel field of research in which large-scale neuroimaging readouts are used as a probe of network activity and dysfunction across species. It is really great pleasure to have Dr. Gozzi here, and without further ado, Dr. Gozzi, I will let you to take it from here. Thank you.
Thank you, Armin. Thank you all for having me here, and yeah, it's great to have the chance to share what we've been doing. And so basically, what I'm going to talk about today is a bit of a summary of work that we've been carrying out over the past 5 years or so, probably even more, actually, some of this work. And this work was aimed at trying to understand what the molecular, what the physiological basis of network dysfunction that we can measure with fMRI are in complex brain disorders. And this is where it all started. But I would like to begin with a couple of clarifications. And the first one is a terminology alert. Before we dig into the science, and it is the terminology related to this word here, which is a bit of something that we kind of came up with. So the alert here is related to the fact that with this connectivity, I don't refer, throughout my talk, I won't be referring to disconnection or separation, but this is just like a shortening of dysfunctional connectivity. So this may indicate either increased or decreased connectivity. Okay, so it's just a label to refer to connectivity alteration. And the second clarification, which is perhaps a bit of a spoiler, but it's an important point I want to make at the beginning, is that this presentation here is not necessarily about brain disorders and disruption of connectivity. And as a matter of fact, some of the mechanisms that we discovered in the process that and I'm going to show you and explain them well enough, I hope, are really generalizable, and they actually apply to the healthy brain. So this is something I would like to say, because of course I will start, and I'll show you. I'm gonna start with the menu very sure, momentarily. I want to show you what we're going to talk about, but the goal is to use our research. It's a bit of a journey. Our research on the mechanism disruption of connectivity, to see where we got in terms of understanding of the basis, okay, the physiological basis of this phenomenon in the healthy brain. So do not be put off or tune out if I have this the occasional reference to disease, because this is really important. In the end, if you bear with me, possibly you will gather, I hope, interesting information. Okay, so that's a bit of a menu of my talk, and there are two parts. In the first one, which I try to keep short, I would like to show you some effort that we've been carrying out once again, over the past 5 years or so, aimed at try and decode the molecular basis of alterations of fMRI connectivity, alterations that are associated with complex disorders. Specifically, in this case, it's autism. But as you can see here, I put another disclaimer. So this is not about autism. And if you guys are not interested in autism, that's fine, because you can just focus on the on the paradigm, okay? And it apply to different, for example, disorders or conditions. And there is no need to know what autism is like. I will explain just the two key facts you need to have in mind. And then the second part, building on the work we've been doing on initially, and the first part of my talk, in the second part, I will try to explain how we can physiologically decode the signals that we found to be altered, what basically what's their meaning in terms of physiology. Okay. And since I assume that most of you don't know me, let me just also, before I get I dig into the science, let me just explain a little where we come from. Well, as Armin said, yeah, we're interested in investigating the functional organization of the mammalian brain, and I think there's now some consensus that if you want to understand mechanistically a complex phenomenon such as functional connectivity, whether it is with fMRI or other methods, we need to try and complement clinical approaches with methods that allow to establish causality between events. And so in order to fill this sort of gap that we have, right, between models and investigations for brain function at different levels, what we pioneered a few years back was basically to try and implement network level measurements of activity in physiologically accessible species. In our case, it's the mouse. And clearly, given that we want our research to be relevant also for humans, right, for human investigations, here we decided to use in rodents a readout, a measure of network activity, which is resting state fMRI, which is convenient because we can somehow, of course, with many caveats, we have to be careful, but we can translate it across species and without having just to make inferences about what really is the correlate to both. It's the same biophysical phenomenon. Okay, and so this is what pioneered this field really. If you got back in 10 years ago, and basically now with this platform, we can manipulate connectivity, have like multimodal measurements. And as part of our initial research, what is really emerged is that notwithstanding the large evolutionary separation between rodents and humans, which is around 100 million years or so, there are some correspondences also in the how these networks are organized. For example, rodents have default mode network, they have salience network. So, I mean, of course, with a lot of caution and a lot of caveats, we can perhaps also extrapolate changes across species. And I'm going to show you an example later. Okay. All right. So the first point, the first part of my talk is about the molecular determinants of connectivity. I have to start here. And as I said, I'm going to talk a little bit about autism. And it's not important you are autism expert. The only thing you need to know is refresher is that autism is diagnosed behaviorally, right? Like many other complex disorders, neuropsychiatric disorders. And the second important point is that, of course, it's early developmental disorders where, that's pretty much around 1 out of 100 children, pretty much. And well, I mean, I think the reason why we got interested in autism is because it encapsulates, it epitomizes the sort of the field of research in autism. It epitomizes really the standstill we have encountered when it comes to applying, for example, fMRI connectivity, system level measurements of network activity in complex disorders. And what I refer to is to give you more context, is the fact that I'm pretty sure that many people here that perhaps will listen to my talk, like 10 years ago, 20 years ago, when these measures were beginning to be implemented in clinical populations, in humans, had like this sort of inconfessable desire, right, to use these methods, like think of fMRI connectivity, to address this question here. Can we see autism in the brain? And this means basically two things. It may mean more, but two things particular, at least to me. When I say, can I see autism, I have in mind, for example, like, hopefully, like some sort of a better diagnosis, right? Perhaps a network dysfunction that is specific to autism, and we can see earlier, or we can use to differentiate autism from other disorders. And perhaps also if you were able to identify this, this specific network alteration that is specific to autism, maybe we can also understand what the pathology is about, right? You know, what circuits are disrupted and so on and so forth. And so this really prompted a lot of studies over the years using, for example, fMRI connectivity in autism. And the results we got are summarized here, and I think they're very interesting. And they're very interesting because there's a lot of evidence of decreased connectivity, right? But it is also interesting because there's a lot of evidence of increased connectivity. Even more interesting, perhaps, or equally interesting, that there are also studies showing both decreased and increased connectivity. And so if you go back to our question here, can we see autism? The answer is not yet. We cannot see it, okay? There's no way we can see it because there's so much heterogeneity and variability. Okay, and so what is really interesting, however, and what I found really fascinating when I started this research in this field was that when I was reading these papers, I realized that there is still a fraction of the, especially clinical population, that thinks this way. Basically, the idea you can still read in some papers is, okay, now there must be a signature of dysfunction that is specific to autism, but the reason we have not found it is because we've been reusing the wrong tools. And specifically, we're talking about fMRI here. You know, fMRI is always like vituperated, the reproducibility crisis, and so on and so forth. And so basically, according to many, this, what I'm showing you here, is a spectacular manifestation of the reproducibility crisis in brain imaging. However, there's an equally probable or likely plausible, I would say, possibility. And that's really where our research started. And the second alternative hypothesis is that we will not seen autism so far is because there is no such thing as a single specific network dysfunction that is, appreciable and measurable in autism. And and this is in fact, I mean, I'm a neurobiologist. This is in fact in line with the neurobiologists of autism. For example, there's so much heterogeneity in the comorbidity, but more important, and this is a point I want to deliver just for you to understand. As you probably know, autism is strongly genetic, strongly heritable. But when you look at the genetics of autism, there is no convergence on specific mechanisms. For example, there have been like more now than 100 high confidence autism risk mutations identified, none of which explains more than 1% of cases. And so it's really right spread out in terms of, and in fact, it is a spectrum, right? And so we thought, okay, so how can we rigorously test these alternative hypotheses? There are different signatures. Well, in humans, this is difficult to be done. Why? First of all, because for most people, there is no genetic testing. We don't know, the underlying biology, and also because there's so much variability also in humans, right? In even healthy populations, and both in terms also environmental contributions and the underlying genetics, right, which is different. And so what we thought is, okay, if it can be done in humans, maybe it can be done in mice. And so what we thought is, okay, let's try to leverage imaging genetics in the mouse. And the idea is very simple. Basically, autism is strongly genetics. That's what I told you. But you also know that genetics is very important in shaping our functional connectome. There are so many studies out there. And so we thought we could use genetic heterogeneity as a key for an etiological decoding of autism. The idea here is that, for example, mouse models that have the same mutations should have pretty much, like more homogeneous changes compared to others. So we can use different models, different mutations as tiles of a puzzle. And we can see whether we can, build like a landscape or whether they overlap. And the big advantage of using rodents are, well, first of all, in rodents, or that we have a clear control population. We can control very well genetics, right? Because these mutants are like clones, except for a single mutation in a single gene. And the second thing is that you can control very well the environmental variability as well, which in humans is difficult to do. We can control also head motion very well, right? We can fix the head or use like less sedation, which is another plague of human imaging, especially for psychiatry imaging or developmental neurology. And so that's what we thought. Okay, so together we amassed like more than 20 mouse lines in collaboration with Zurich, and we map connectivity changes for each of these mutations. And the results I'm going to show you here are basically summary what we found. And here there's some sort of color coding, so keep that in mind, okay? Because this color coding is going to stay throughout the presentation. And blue means reduced fMRI connectivity, okay? This is weighted degree centrality, so it's like a summary measure. And so each of these, like this is the top view of the mouse brain. Each brain basically is the difference in connectivity produced by a single mutation. So there are different ones, okay, here. And as you can see, there are in fact some convergences, right? For example, these are all blue, and there is the blue tends to be on this midline, which is kind of the default mode network of the mouse. However, if we zoom out, if we add mutations, we see there's a shift. This is more like sensory. There are some mutations for which there is pretty much nothing in the cortex. There may be stuff subcortical. I'm not plotting it here. And then there's my favorite group, which is this one here. And it is my favorite group because it goes from blue to red. And red, I'm sure you guessed it, is the opposite. It's like increased connectivity. And the thing I like this is because I think this gives you an idea of what's going on in autism, right? You know, blue and red cancel each other out, right? So if there were like people, and if I was just scanning randomly, you know, it's not surprising that sometimes you get more blue, sometimes you get more red, sometimes you get a bit of both or nothing. Okay, and so just translating this in biological terms, this indicates that etiological, in this case, it's mostly genetic, but there are also immune systems here, immune alteration, sorry. So etiological variability is a key determinant of fMRI connectivity alteration that we observe possibly in humans, certainly in the mouse. Okay, and so perhaps we should stop, chasing this elusive specific signature of autism, and we should instead focus on finding how many autisms we have, right? Because here, what is really interesting, if you focus on the divergence, is that perhaps when there are some homogeneities, like in terms of distribution, there might be, and this is something we have to test, and we tested, there might be some like common biology supporting these convergences, okay? And so that's really the next big question. So why are red red? Why are blue blue? And are these expression of different pathological mechanisms? And are these pathological mechanisms, can they be grouped into some macro categories that can allow us really to define subgroups, okay? Well, this can be done rather easily in the mouse with this same database. How? Well, I can identify some subtypes, okay? And I can, for each of them, I have like this tag, which is the gene, and I can see if these genes are part of like some biologically homogeneous pathway. And that's something we can do. And the first question, however, is okay. Let's see how many groups can we identify. Well, here we can use like machine learning, of course, but and we did use. I think we used k-means. If k-means is machine learning, I'm not sure. But basically, what I would like to tell you here is that here we made like a strategic decision. The strategic decision is okay. We don't want to go like too noisy, too small group. We don't want to focus on groups that are too small. Okay? And here we want to test like a general hypothesis. Red should be different and dissociable from blue. Because if you cannot, in terms of biology, etiology, if we cannot tell these apart, then we are in trouble, right? Because these must be different. These effects are really, one the opposite of the other. And so we basically start with two groups. We generated like the average like disconnectivity map. So it's like a bit of a grand average of the red here on the blue. And these are interesting priors that show you hotspots of disconnectivity alterations. And these are going to come in useful later, by the way. And so we have these two groups. Let's go back to our questions. Are these two groups associated to different dissociable mechanisms? Well, this can be done easily in silico. You can put all the genes we have right into like a silico system. We can see how these genes interact. We generate the interactome, and then we can do what's called like a gene ontology. We can see whether there are pathways, okay? And to cut this long story short, this is hyperconnected, this is hyperconnected. You can see they are highly dissociable. So hyperconnectivity, it is strongly associated with synaptic signaling. Okay, so genes that are involved in that, while hyperconnected more on transcriptional dysregulation and immune system alteration. So we have some sort of like dissociation here. Okay, this is probably coarse at this stage, but this is where we want it to be. And that's, however, well, that's probably interesting, but this is mouse studies, and mice do not have autism. Okay, this is, I'm all aware of this, but mice can be used as tools for understanding what's the role of genetics on these of immune mechanisms in altering network activity. And so the big question I'm sure many of you have is, does this matter at all for humans? Okay. And so can we identify these kind of subtypes in humans? And here I have to make an important say like clarification, if you like, or make a digression, which is important. In the mouse, I told you, we can kind of recreate the genetic landscape of autism, but we cannot replicate the whole genetic background of autism. Why? Well, this is a little technical, but it's very simple. So it depends on the study, but the largest fraction of autism it is still genetic somehow, but it is supported by common variants. So variants, genetic variants that are present in the population, many of them, they produce like, so maybe you heard polygenic risk score. And so they give you like the susceptibility to develop autism. And so what we can model well in mice is not as common variants, because clearly we need like to have mice with many small mutations, but we can model very well what are called rare variants, which are the ones that produce stronger symptoms, like more severe symptoms, and there are the ones that can be modeled well in humans, and then in rodents, and normally they are like a mutation in a single gene, for example. And so why am I telling you this? Well, because of course, if you try to translate mouse to human, we do not expect to decode the whole landscape, but we have in mind something different. We think that perhaps some subtypes in humans can be related to the pathways that are modeled and disrupted in mice. We don't expect to have like a whole explanation. Okay, and okay, so how can we try and translate this, what we found in the mouse? This is like a summary. Okay, what does that show you? Models, subtypes, and then we do molecular pathways. How can we translate this in humans? Well, this is kind of, well, it's conceptually easy, because in humans we have these large databases. One is called ABIDE, some of you might have heard it, of scans in typically developing and autistic people. And what we thought is, okay, let's do this. Let's use our priors, so the hotspots of this connectivity, to guide a selection of scans in humans that have similar features, so that are similarly hypoconnected in those same regions that we found in the mouse or human equivalent, compared to typical developing populations. And the same with the type 2, hyper in the same regions. Okay, we see if there are those scans, and then we try somehow to figure out a way to see whether we can link those to molecular mechanisms. And okay, so to cut very long story short, I want to just to get to the point now. We do find those scans, okay, first of all, which is something that, it's not to be taken for granted. We have some scans like around 7%, 16% are this hyperconnectivity kind of widespread. It's interesting because together they kind of make this 20%, 25 I told you before. So far it's so good. And now the question is, are these real subtypes, right? Dissociable. Okay, if they are, they must be present in pretty much all the sites that are part of this ABIDE dataset, and that's the case. Look at that. All of them, they have a fraction of these, okay? And here we can also see that there are some AMSI type population, as I told you, and we are okay with that, okay? All right, so if these are two groups, subtypes that can be, differentiated, they should be replicable, and in fact they are replicable, okay? Because we discovery, replication, and they are reproducing. And once again, 30% pretty much of the whole sample. These two subgroups are also dissociable in terms of their network involvement. Okay, they are not really like one the mirror of each other. Going back, this is not like one the mirror of this, okay, red and blue, but they kind of affect different areas. And they also, okay, I don't want to make a big story out of this, but it is interesting just to note there are also slightly but significantly different in terms of severity of symptoms. But maybe, I mean, what is really more important here, besides, getting into the clinical relevance of this product, perhaps we discuss later. The most important question is really the proof of concept. Can we just, demonstrate that what is the models that we found in mice, so really this pathway that are altered can be retraced and decoded in humans? Well, here we need to address this question. We need some molecular, right? Sort of signature, some molecular evidence. What we did here is something that many other groups have been using. So we use what we call gene decoding. So we use the Allen Brain Institute post-mortem atlas. We can make a list of genes that are specially enriched in the same areas that are hyper or hypoconnected. We make this list and we decode it with gene ontology. And what we found, there are two interesting findings. The first one is that the genes that we decode that have the same, spatial pattern, so to speak, are strongly enriched in genes that are differentially expressed in autism post-mortem. So this like enrichment for genes that are important for autism is very, very high. And what's really also interesting is that when we try to use this like ontology decoding of hyperconnectivity of hyper, we have synaptic signaling in hyperconnectivity and immune system in hyperconnectivity. If you probably remember, this is exactly what we found in mice. Okay, so these ontology terms are really the ones that are more important for hyperconnectivity in humans. They're also more important for mice. And this is okay using two different gene decoding methods. And so to cut a long story short, again, to get to the end of this first part, by using this approach, we were able, starting from some sort of like I use quote ground truth in rodents, to identify two dominant hypo and hyperconnectivity subtypes in autism. One that seems to be related to synaptic alterations, the other one to immune-related genes. In mice, it's also transcriptional, and this accounts for around 30% of autistic cases. And this is, I think, nice because it underscores the idea that the etiology behind these disorders can be behind the variability we see in humans. And perhaps, once again, as a field, we should try and understand how we can decode and make the most of this, okay? There's a question there, probably I'll keep it for later, if at all possible. And I would like to try to move on to the second part. Or am I supposed to address it now? You let me know, Armin. Okay, I go ahead, okay?
Sure. Yes. Yes. Please go ahead. Thank you.
Yeah, we'll keep it for the end of discussion. Okay. So far so good. I hope. Now the question, and this is when we move a little bit into like what's the physiology? Because I mean, okay, hyperconnectivity, they're like these vague terms. What does it even mean, right? So what's the physiology behind this? What's happening? And okay, so that's something that we've been investigating in the lab. And the question here, of course, was where do we begin, right? This is again the representation what I showed you before. So if you want to test this, what does hyperconnectivity, hyper mean? Where do you start? Well, we start again from what I just been showing you. Synaptic signaling is the dysfunction that was associated with the hyperconnectivity. And that's interesting because in complex disorders, alterations in synaptic signal, for example, like especially early developmental disorders like autism, synaptic dysfunction means mostly, clearly not exclusively, but mostly one thing. It means excitation inhibition imbalance. And namely, it means increased excitability because autism is strongly comorbid with, it's got high comorbidity with epilepsy. Basically one kid out of three have epilepsy. And so we said, okay, maybe that's an interesting clue, right? And so that's where our like manipulations began. We said, okay, let's do something. Let's test this hypothesis. And this can be done by using chemogenetics. Chemogenetics like optogenetics, and instead of using light to activate neurons, you can dial up or dial down neuronal activity, okay? And we did it in the medial prefrontal cortex, and we said, okay, let's try to increase excitability and see what we get, okay? And this can be done two ways. And I call it here increased excitation, which means exciting pyramidal neurons, or it can be done by reducing inhibition. And I call it here reduce inhibition. Here what we did, we inhibited fast spiking neurons, parvalbumin positive. Okay? And clearly, if you want to make sure that what we're doing here makes sense, we need to use electrophysiology, right? Because we want proof that we are increasing excitability. So from now on, I'm going to show you both electrophysiology and MRI. Okay, so these are like our first analysis, kind of sanity checks. So when I use the manipulation, I call increased excitation, we have more firing. This is like multi-unit activity, firing. These neurons are firing more. And as you can see, the zero is when I give this drug that increases excitability. The chemogenetic actuator is called CNO. And you see it goes up. And the same is with I reduce inhibition. That's more firing. So good, because that's exactly what we want to see here. What about the spectral properties of these electrophysiological activity? That's another interesting thing, right? And when we look at that, this is indeed very interesting because when the two spectra, right, are very different. And this is the spectrum of electrophysiology in the manipulated area, which is prefrontal cortex, well, medial prefrontal cortex, anterior cingulate. You see how different they are. When you increase excitation, you have a strong suppression of oscillatory activity and you have high frequency firing. Okay, so these neurons are firing asynchronously. When you increase inhibition, when you reduce inhibition, you have a strong increase in oscillatory activity. You can see the frequency bands here, okay, and which is totally different. Okay, so what about bold fMRI connectivity? Well, first of all, because I don't want to confuse you, let me show you locally bold what's happening to bold signal. And this is kind of predictable because there's this beautiful work that was pioneered by Logothitis and others showing that bold activity is very sensitive to high frequency activity. And as expected, when I use those manipulations, increase excitation, this is the bold signal going up in the medial prefrontal cortex. You see how strong it is. You can map it. It is happening where it should happen because this is the prefrontal cortex, and this is before, and then we have like this increase. And the same is happening when you reduce inhibition, which is also cool because that's what you expect. I show you there's a lot of oscillatory activity, but it is high frequency, that's high gamma, and then you have more bold here in the prefrontal cortex. Okay, and signal goes up. But what about the fMRI connectivity? That's exactly what we're interested in. Okay. Well, the results we get with connectivity are, I think, puzzling, perhaps, or surprising, and interesting at the same time. Why? Well, this is what we found. Again, this is a seed-based analysis of the manipulated regions, control. When you look at increased excitation, basically you don't have anything. So the difference is a strong blue. So you have a strong decrease in fMRI connectivity. And this is, of course, interesting, but the puzzling part, if you like, is that you get the same when you do reduce inhibition. Again, you have blue, you have reduced connectivity. And this is right, perhaps a little puzzling and surprising because, first of all, I just show you, like one slide before, that we are increasing the activity in this region, yet connectivity is going down. And also, I told you that these two regions have different spectral properties, and yet they both produce hyperconnectivity. So that's difficult, right, to reconcile. However, there's also an interesting angle here, and the interesting angle is that these two manipulations increase excitability, and so maybe there's a bit of a general relationship here, which is if I increase excitability of one region, I have lower connectivity with the region between the region and its terminals. This is like a possibility, and if this actual possibility is true, it's valid, it comes with a prediction, which is if I do the opposite, if I reduce excitation, so basically if I silence a region, I should have more connectivity. And that's exactly what we get. And this is something that we can test very easily because, in fact, this is published data. In this study here, what we did, we used another chemogenetic manipulation. In this case, that dials down neuronal activity. You can see multi-unit activity. Okay, it's got its own spectral activity. What's important, however, it is suppressing beta and gamma, so high frequency. And when you look at connectivity, connectivity goes up and it becomes red. And that's an interesting point, in my opinion, because here, this is suggesting a general sort of relationship, which is more excitability, more firing, at least in the cortex or in the frontal cortex, lower connectivity, and the other way around. But what about the puzzling part? How can we reconcile this data with a different spectral signature? Well, there is an important angle here that we have to remember, one important fact. And I'm sure many of you know this, but I keep repeating this when I present this work, which is we shouldn't forget that despite its name, fMRI connectivity is not a measure of connectivity, it's a measure of synchronicity between signals across regions, right? And so we said, okay, if you want to understand what happens here, we need to measure synchrony across regions. So what we did here, we implemented two electrode electrophysiology. One, this is the manipulated area, and this is one an area that's a posterior cingulate, strongly innervated. And this is a key graph that I would like you to understand. So I'll try to explain very well. This is LFP coherency, so a measure of connectivity between regions. Okay, how coupled they are. Okay, I could have used even Pearson correlation, but coherency probably is better because it's sensitive also to the phase. That's a technicality. And that's the coherency modulation, which means this is the difference in coherency produced by the two manipulations. So zero means no difference. But as you can see, when I induce the excitation, there is a difference between with zero, right? It's got its own profile, and especially when I reduce inhibition, coherency goes up here. And you can see the band. So delta, beta, whatever. And so, I mean, without getting too deep into the details, you can see that the two coherency. So this is like a EEG connectivity, right? Only that it's intracortical. So coherency is very different between the two manipulations. Yet they both produce hypoconnectivity. So why is that? What's happening here? Well, one way we can explain this result is by assuming, is by hypothesizing that fMRI connectivity is disproportionately sensitive to the very low coupling frequencies. Okay, so basically to this part here of the spectrum, which I'm going to show you here. And this is again the same, okay, same coherency, but it's log. Okay, and if this hypothesis is correct, we expect in this case they go connectivity goes down, right? So we expect this to be below zero, and they are below zero actually, which is interesting. So this could be one way to somehow reconcile, and this is something that makes sense, right? Because fMRI, it is indeed a slow phenomenon, right? And if this is correct, if this hypothesis makes sense, actually, then it comes with another prediction, which is, you might remember I showed you 1 minute ago, 2 minutes ago, that if I silence a region, I have more fMRI connectivity. And so with silencing, I should have something happening here, right? I should have stronger coherency here, and perhaps a lot of other things in other bands. But our hypothesis here that fMRI is very sensitive or disproportionately sensitive to this. And that's exactly the case, okay? As expected, we have stronger coupling here, and there's other stuff happening here, but our hypothesis is that what matters is this. And there's another prediction again that comes with this model. And the idea here is that if this is what matters, the change in fMRI produced by our manipulations should covary its magnitude with the magnitude of this coherence, right? In these bands, but not in the others. And that's exactly what we see. You see there's this relationship across manipulations, and the relationship where we have significance, or sorry, the bands where we have significance is 0, 1.4 hertz, and slow is lower than 1 hertz. This is like a split up. But if you go higher, you don't see much, okay? And so this is interesting, I think, I hope, or we find this potentially interesting, because it suggests a simple model, right? When you want to relate fMRI connectivity with electrophysiology, that fMRI connectivity is disproportionately sensitive, is dominated by coupling in these low rhythms. And this I mean, okay, maybe it's obvious to some, but it's not that obvious, right? Especially when you begin to use a bit more of like a causal approach. And so I want to use like 5 more minutes, if possible, to show you something else, something even something new that we've been trying to implement to prove this point here. Okay, thanks. Because okay, so far so good. I would say okay, there's this interesting relationship, but it is still correlation, right? Correlation between bands. So we thought, okay, can we just go a little further and prove what I'm saying here? That fMRI is really sensitive to slow rhythms and kind of insensitive or less sensitive to faster rhythms. Which also comes to the question of how do fast rhythms influence these signals? And I'll keep this for for the last slide, basically. And so we said, okay, let's try to go here in a more like causal section. And so what we thought is, okay, this can be done at least conceptually. Technically, it's kind of complicated, but conceptually easily in the mouse, because you can use optogenetics to generate synthetic oscillations, right, in the mouse, and we can concurrently record the fMRI. Okay. And so what we did here, we can modulate the hub region of the, okay, sorry, this is just a repetition. We modulated the medial prefrontal cortex. Okay, that's what I've been explaining to you. And here we use optogenetics. Optogenetics are flashes or lights that can be used to produce rhythmic activity. And clearly, given that we want to produce rhythms in the brain, the living brain, of course, we don't want to rewrite physiology books. We know that most rhythms in the brain are nested rhythms. They have a carrier frequency and they have an envelope. Envelopes like changes in the power, okay, of this carrier frequency. And so here we use something that was very convenient for us because of the optogenetics method. So we use a pretty fast carrier, 20 hertz. I think it's beta, if I'm not wrong. And then does this, and we change its envelope slowly, okay, 0.1 hertz, which is infra slow. And what I'm showing you here is sanity check, electrophysiology. You can see that we can get this very slow activity, carrier and slow envelope. And when we measure with electrophysiology, the time locked to 0.1 hertz in the prefrontal cortex, it's as expected around 100%. So most of the activity there is entrained to this synthetic artificial rhythm. Okay, so what happens to fMRI connectivity when we do this? Well, this is the connectome, the axonal connectome of the medial prefrontal cortex. Okay, and these are also, this is just the Allen-Brain Institute connector, so axons. This is obtained with viruses. And so this is just our reference, okay, because we are exciting, like producing a rhythm here, and so this rhythm is supposed to propagate at least in these regions. And this is the corresponding connectivity. Okay, and red, by now it's increased, so we have a strong increase in fMRI connectivity, very strong. We get highly significant. And there is also something, okay, this is interesting to note. However, not in all the innervated regions. For example, this is the striatum, for those who are wondering, and there is no increased connectivity here. Why? Well, this is more of a problem of Pearson correlation than anything else. Because if you measure phase locking, okay, of the fMRI signal with this synthetic infraslow fluctuation, we see that it is strongly tight, coupled where it should be. And this is, if you like, a nice sanity check. So infraslow activity is very important in supporting fMRI connectivity, okay, fMRI coupling across the whole brain. And really the bullet, the proof is this one here, right? You can see that the phase locking concentration is very strong where it should be. Oh, okay, but the question is, what about faster rhythms then? Okay, I told you that here we use like a carrier frequency which was fast. So there's a carrier frequency, there's the infraslow. And so which one of the two is more important? Well, here at the beginning we were discussing the lab. Well, let's just vary the carrier frequency, in I don't know, from delta to beta, even gamma. But that's too much work because this is a complex experiment, and so we took a shortcut. Here we said, okay, if the carrier frequency is important, if we scramble it, if we dither it, and that's what we did here, you can see, if we scramble it, right, the coupling, we see the fMRI connectivity should change because this is just random. We don't have like a carrier of of beta. This is milliseconds, okay, between pulses. It is all, random. And once again, just to summarize, as you can see here, and here there's also proper statistical testing behind this, we don't see any difference, basically. And that's interesting because this suggests that what is, as I hypothesized, what is really producing this entrainment is the envelope, is not the carrier frequency. Okay? But what about a faster envelope? Can I show you the envelope I show you? So this slow modulation was 0.1. What if we go 4 hertz? And we can go 4 hertz because of the help also my friend Noam here that helped us set up 75 millisecond single slice fMRI. Okay, and so we said, okay, let's use an envelope at 4 hertz. This is electrophysiology. It is showing that the rhythm is strongly training like firing in this region. But when I do fMRI connectivity, this is our reference, 0.1 hertz. You can see phase locking very strong. I vary the envelope at 4 hertz and I don't see anything, zero. After this coupling at the large scale is not happening. This is just too fast. Okay. And this is again a nice, I think, demonstration of what I was telling you before. fMRI connectivity is primarily sensitive to changes in this very slow frequency. 4 hertz, it's already too fast. Okay. And so the question you might, let me see what's next. Yeah, exactly. So basically, pretty much the last slide. So the model we propose is this one here, okay? And I think I've been repeating this over and over, but fMRI connectivity is dominated by slow rhythms. It's, remember, very sensitive to those, okay? I'm not there to say it is totally insensitive to others, but based on our data, the contribution of the rhythms is really negligible. And then this rule here, which is also important, okay? Cortical excitability inversely modulates fMRI connectivity. So you have this like more is less and less is more. More excitability, lower connectivity. How? Well, how is just, by interactions between the excitability, this high frequency activity with these low rhythms, right? So that's pretty much what we are supporting here, what we are bringing forward as a model. That faster neural activity is important. Of course, it can affect fMRI connectivity, but it does insofar it interacts with slow activity, okay? And one interaction is when one region is very excitable, and so this very less localized slow activity interacts locally, right, with this fast activity, and you have like some sort of interference. So that's the model we are proposing. I think I have probably, yeah. So that's pretty much it, okay? And so we started here, and now we can add this tentative for sure, additional explanation. So when we have hyperconnectivity, not necessarily noticed, we might have like a state of hyperexcitability. And when we have hyperconnectivity, you have the opposite, perhaps too much inhibition, neuronal loss. Okay. And so I think I'm done here. And so just last messages. Well, I think I hope I made clear the cross-species approaches are useful and can help us understand what happens in terms of connectivity alterations. I think our data suggests that intrinsic fMRI connectivity is supported by slow electrophysiological coupling, or is sensitive to, if you like, perhaps not supported. And there's this general rule that we seem to see, especially in the cortex, because we didn't test it other regions and it could be different, where neuronal excitability inversely modulates fMRI connectivity via interactions with infraslow activity. And I think that's it. I am really to thank the amazing lab I have. I'm very lucky to have such talented people. Also, of course, all the funding agencies. And yeah, we're also hiring, you guys know, people who want to come this way of Europe. We are positioned both at postdoctoral PhD and laboratory technicians as well. Thank you for your attention. And if there are questions, very happy to have a discussion.
Thank you so much. It was a wonderful talk, a lot of information. So I start with my question. I take advantage of my role. There are, we know, like at least from fMRI study, that when we have like a connectivity being driven with those high activity in two time courses that are coupling. And also, like if we pick the like a region of, for example, PCC, and we find like a the region, the time point that have a high activity, we can literally like see, for example, the formal network. We can do the same thing for other network. So I was wondering if you can comment on those observations that high activity are linked to high connectivity and these findings, please. Thank you.
That's it's a very interesting question, and I don't have like off the top of my head a real explanation for that. Clearly, I mean there are different possible angles here to explain this the contribution of this burst of activity. Here, let's start from what is one of the limitations of what I'm suggesting, or the model I'm suggesting here. Well, there are many, of course, and one is that it is oversimplistic, right? We are just, there's very simple rules. And it is oversimplistic in a way that, for example, it doesn't account for fluctuations in arousal, which are clearly emerging as an important driver also of coupling between regions. So what's the meaning of this burst of activities, these peaks of high activity? I don't know. Some people are discussing them in the context of arousal peaks that can be related to either intrinsic or other causes. In our model, this is used. What I've been showing you is with light sedation. So we can have a state that is kind of stable, and that's really a very basic well, and of course even with sedation we have these peaks, however, because our work we found, for example, using coactivation patterns, that we have like these peaks of activity. Yeah, I don't know really. As you can tell, I've been trying to fumble around, but I don't have a clear explanation for those. No, that's clearly important, where they come from, whether this interaction for this flow ongoing activity with peaks of activity, local, I don't know. Yeah, that's an important question to look into.
Thank you. It's very interesting works. Yeah. So I will move and ask, read some of the questions that have been shared in chat. So Saris is asking, in some subjects have a more bold variation and others have a very small variation in bold, like in general. He's asking, do you know if there might be related to the genetic expression, or you have any comment on that?
It could very well be. It can be multifactorial. I don't think we can, based on this work, make any firm assumption about what's happening. That's a lot. There are many sources of variation, and I don't think we are in a position where we can really attribute what we see to one or another. And yeah, this is, as you say, as I try to deliver, it's really like proof of concept work that hopefully will move a little bit the field forward, right, to some schema. I mean, for example, many people still find it difficult to understand. Many people, when I present this story, they find very counterintuitive the idea that if you silence a region, you have more connectivity. But if you consider it in the context of oscillations, it's not so counterintuitive. So, yeah, once again, I don't have an explanation or a theory for this.
Thank you. I believe like these work provide a very good foundation to help us better understand for sure. And another question from Gopi, he's asking, has the link between fMRI connectivity and excitatory inhibitory been shown in human using MRS or not?
Not with MRS, but that I know, not that I know of, but that's work from our lab, and you may want to look it up. It's work we published 2 years ago in eLife, and I'm not sure I have a slide. Yeah, I do actually. If I quickly can, I don't know. I stopped sharing because I wanted to see in the face. Okay, sorry. I'll try to share it very quick. Can you see my screen?
Thank you.
Yeah.
Yes.
Yeah. So this, sorry. This is the work we published. So basically here, there are ways of relating, and we have 2 follow-up papers, yeah, they're not papers yet, but hopefully soon, that are building on this story. And basically, this is in collaboration with Stefano, who's a mathematician, and Mike. And here we can try to link regional activity with fMRI to a parameter that is called Hurst index, which probably some of you, if you have physics, know what it is, which is like the slope of the 1/2 spectrum of the signal. And here, using biophysical modeling, so basically the same manipulation I show you with rats, we found that the first index is different and sensitive to local excitability. And here we have a proof of concept study where in autism we find that you can check out the paper we validated extensively, basically. But we linked some of these parameters also to genetic alterations. A little bit what on the line I've been showing you. So yes, elaborate answer, which can be summarized as yes, there are some means like this, check it out, the code is there. And we have improved now estimates, which should be able to release in the new year. So to some extent, I think it is possible.
Thank you. Thank you very much. If you don't mind, I will ask one more question before I let you go. I know it's very late there. So, thank you. Cyrus is asking if there is any relationship between 4 Hertz in the low field potential and the 4 Hertz that we are seeing in EEG and MEG.
Well, I surely hope so, yes. Yeah, pretty much the same. Yeah, of course, they are the same rhythms. Okay, there are modality-specific differences in the sensitivity of the signals. Like we know that with MEG, if you call, well, okay, I don't want to be too technical. If you look at the correlation of these power spectra in MEG with fMRI or EEG, there are kind of differences. For example, in MEG is more on the lower rhythms for some reasons, and in EEG is on faster. But yes, of course, I think this is exactly the idea. What we did here was just to measure intracortically, so like with an electrode. So it's a little bit like a NICO thing. Yeah, of course, the rhythms are clearly translatable, so that's something to be kept in mind. Another important message here, however, is if you want to differentiate excitation, increased excitation for reduced inhibition, you should use EEG perhaps, and not fMRI. During fMRI, they converge.
Thank you.